Assalamu alaikum. We will continue our lectures about skin grafting. We go to the half of the reconstructive letters. We consider this is reconstructive letters with the simple one to the more complicated whenever we go upward. So the mid of these letters, we consider the skin graft. The skin graft, which consists piece of the skin, consists of the epidermis and some portion of the dermis. So it is not only epidermis, it's some portion of the dermis removal from the its blood supply, from its blood supply and transfer to the another location. This is the definition for the skin graft. So what composition of this skin graft? It's a epidermis and portion of the dermis. And this dermis thickness depend on the type of the skin graft, whether full thickness, whether partial thickness, skin graft. Location of this skin graft, we can consider this is historical elements which is the Revidians first skin graft done by the one of the surgeon Reverdine, and Pollux describe and do the autograft frames from the same person to one place to another place, which is the more common type of the skin graft. Thirsch define the thin skin graft, which is the split skin graft, Thirsch graft. And Wolf describe the full thickness skin graft, so we can term that Wolf graft, which is the full thickness skin graft. This is historical elements. Uses, where can we use the skin graft? To close any wound with good blood supply. There is important things. Defect cannot be closed primarily, but where the area have a good blood supply. As element of temporary closures, we sometimes require, some place required only temporary close it, not as if a primary full closure, only temporary for a period of time. Controlled site infection, there's have a lot of infection in this place, cannot be closed immediately, and we can get abscess collections and can be do machine skin graft and put it to control the infection. Anesthetic replacement, asthenic replacement means there is element of the element of beauty to avoid the ugly appearance of the area. So asthenic replacement, closure of the donor site, like donor site for the flap. The donor site for the flap can be closed graft, or sometimes the mucosal replacement, and it's covered by the also by the grafting. What is type of this skin graft? It is autograft or allograft or xenograft. Autograft with the same person from one place to another place. Allograft from the human to human. Homograft from the same species, while xenograft from the animals or from the other mammals to or other mammals, other animals. Use this to cover our body of the human with heterogeneous graft, which is commonly temporary elements. Skin graft typing according to the thickness. Thickness we consider according to thickness. If it is thick layer of the dermis, we consider a thick graft, while we consider this is a full graft. While the split skin graft, which is depend on the very thin layer of the dermis, contain variable thickness, depend on the thickness of the skin we will take from it, and also whatever the type of skin graft is contained, not only the dermis, epidermis, it's also contain the adnexial structures like this part or hole, the sweaty glands, sebaceous gland, hair follicles, some element of the vascularities, capillaries, and also depend on the thickness of this graft, how much these appendages of the skin can be contained. So it's depend on the thickness of the graft, also on the place of the taken from the skin graft. Skin graft, where is the donor site? Donor site from any part of the body can be considered as a donor site for the body. Although the color and texture, thickness of the dermis and vascularity are important, and the donor site morbidity vary considerably. So depend on all these parameters to consider this site is more better as a donor site for the skin graft. Skin graft can be like example for that for the clavicular above clavicle is considered for the superior color to matching what the face similarity, like not when we want a graft on the face, we take from the supraclavicular regions, not from the abdomen, not from the thigh. Like the upper lid skin can also be used as a part to provide a small amount of the very thin skin, full thickness skin graft harvested closed primarily, and the abdominal wall, buttocks, thigh are common donor sites for the split skin graft. Commonly for the donor site, we used the thigh, the inner side of the thigh, or sometimes the outer side of the thigh. This is the common side. Why? Because all the time, a lot of times they are hidden by the clothes, or can be used from the upper limbs, from the lateral aspect of the upper limbs, also can be used lateral aspect of the abdomen, from the buttock area, gluteal regions also can be used. There's a lot of places we can be used. There have we have pictures for that. I don't know where it, I think missed our site. Consider that every element, whether it is element of simple skin graft or element of split thickness or full thickness. We take skin graft from one place and then transfer to the place to cover according to its indication. We consider whatever the place indication, how it's survive, how it's become later on as on the place of the receptions success. Whenever the term, we must understand the term of taking. The success rate of the skin grafting, we depend on the term as it is take, not taked. Take means that the skin graft survive and it's grow very well in its place. Initially, whenever we put the skin graft, what happened for it? How it's get its nutrition? First things is depend on the process of the serum inhibitions. Serum inhibitions, like the plant, whenever the plant that we know that how the plant take its or seeds take its nutrient, its water from the surrounding area. It's similar element. This is usually last for about 24-48 hours. What from the surrounding tissue, from the donor side. So consider whenever I put the graft, I must have good blood supply on the donor side. There is no hypovascular donor side. Must be good blood supply at the donor side. After that, there is a fibrin layer formation within the graft and placed in the recipient bed, binding the graft to the bed, and there is absorption of the nutrition to the graft. And this occurs by the capillary actions from the recipient bed, so that there is imbibitions, then capillary actions from the nutrient at the recipient bed. In insulatory phase, the second phase is insulatory phase, in which the recipient and the donor end capillary are aligned, becoming alignment between there is fusions between the recipient and the donor site by the capillaries. This is the process of insulations. the graft is revascularized through the kissing capillaries that become thickening on the whatever the kissing capillaries become revascularized between the two ends of the graft and the bed of the graft. So that, according to that, whenever I have thin graft, the taking rate is very high because this element of a process of nutrition is very good in the thin layer. Whenever I have a thick graft, so that I have more tissue, more cells required for the nutrition, for the survival, so depend on the vascularity of its place. So the taking rate, some element, some little bit lower for the thick graft and depend on its bed and vascularity of its bed. Causes of failure, whatever the graft, what's the cause of the failure? Development of the hematoma below this graft. I have a good blood supply in the place, I clean it, remove the debris, remove necrotic tissue, and leave some element of bleeding at the bed of this graft. So development hematoma, separate, imbibitions will stop and cannot happen. So I will definitely get failure. It's not taken in this place. Inadequate immobilization. The patient move. My graft in the posterior aspect of the body, and the patient move and cause friction at the early as putting of the graft. So definitely there is not taking. Misevaluation of the wound. The wound is not infected, low vascularity, misevaluation. What? We have the epidermal upside down. The epidermis on the side of it means that the epidermis are inside and the dermis outside. This is the upside down because it's easily happened, especially whenever have wide area and many graft we use to cover, sometimes missing. Where is the upside? They are similar or for the graft, which is the more superficial surface. This is the dermis or this is the side of the epidermis. So this is whenever we put in the upside down, definitely failure. Depending position, like we term that in the posterior aspect, in the place of the, like in the back, in the gluteal regions, in the posterior aspect of the limbs, of considers increase the failure rate. Characteristic of the skin graft and what the complication that can happen for the skin graft. One of the important things is the contraction. The contraction of this skin graft, whether it's a primary contraction or secondary contraction. Whenever we take the graft, immediately it's recoiled and it decreases in size. This is considered as a primary contraction. Why? Because of the effect and because of the activity of the elastin in the dermis. More common this with thick graft. More common with more thicker graft, full thickness graft, more susceptible for the primary contractions. While secondary contractions is occur the new other process, it's the effect of the myofibroblast activities. This is the different activities from that. This occur after the donor side taking and the graft taking, and after the time, look, there is reduction in that side. There is contraction in this place. This is occur more where occur in the thin graft. So one opposite another's. Primary contractions occur with the thick flap because there is a lot of element elastin in the graft. While secondary contraction where occur in the late on, delay, and it's occur in the thin flap, in thin graft. And other elements. We consider this is the contraction of the graft. Other element is there is a sweating in this place. Is the sensation of this place of the graft return? This must be understandable. there is still parasitized this area when I do grafting for it, or there is return of the sensation. Is there is a sweaty gland in this place? Yes. The ability of the graft to sweat depend on the number of the gland transfer and the sympathetic re-innervation of this place. So it is depend on the part of the graft and part of the recipient area from the nerve supply. So it can be retained but depend on the factors. Other sense, whatever the sensory elements of this place also can be retained, but depend on the sensory element of the reception bed, and that can be retained. So the full thickness graft will have a great sensory return because have a great availability of the neurolimal sheath that facilitate reattachment and regrowing that neuro what we can rearrangement of the nerve endings between the graft and the donor site, and this regrowth of these element of the peripheral nerves retain the sensation. So more thick, more retain the sensation. Also the hair follicles can also transfer with the graft. Full thickness graft has contain more hair follicles than split thickness graft, and this is the advantage of the full thickness graft, and also advantage of the use it like on the face or another place that required a special cosmetic or special element of the colors. Technique, we depend on the technique of the taking, we depend on the dermatomes really, the dermatome that used for the taking of the graft. There is electrical, there is manual, like Humby knife, the manual dermatomes, electrical dermatomes. We can take indication and contraindication. Sometimes we do meshing of the graft. The graft, when we take it from its place and we put a special machine like this. This is the measures. We term as the measure. We take by the special machine. This is the electrical dermatome. Take from the special site, like we prepare the site for the donor site, and we take the element of split or full thickness skin graft, and we put a special plate, and this plate then passing through a machine. We term as this machine is the measures. So we create many wounds inside this graft, and after that we fill the area of the donor site with these grafts. So by meshing, first things, the advantage of the meshing, I can take a small area from the donor site to cover a very large area because I fenestrate this graft. Also advantage of it, there is no blood no blood can be no blood underline, it can be accumulated because it's meshed, so facilitate very high, but the disadvantage is what some little bit cosmetically lower than a non-meshing graft. This is the slide that previously I want to express to you. Where is the places for the donor site? There's different place, and commonly we use the thigh and the inner side of the arms. This is the common site for the donor site for the separated skin graft, while there is a special site for the full thickness skin graft. Like when I want to full thickness skin graft on the face, I use supraclavicular regions, or some other element like in the fingers or thumb, I use from the wrist regions full thickness skin graft, or sometimes we use from the thigh or from the inguinal regions. Also these are, or posterior to the post-auricular place. All these places can be take a small piece of the tissue with full thickness skin graft and can be closed primarily without primary closure without defect on the graft. شكرا جزيلا. إن شاء الله الجزء القادم من lectures according to the skin flap.